Welcome, everybody, and thank you for joining us today. My name is Megan Fisk-Clements. I'm FOSSE's food science advisor. At FOSSE, the focus is to understand, translate, and intervene with food allergies. And a key question that our research aims to address is, why has there been such a rapid increase in food allergies over the last few decades? So last month, we introduced our new seminar series, Project Safe, the Science of Adjuvants in Food and the Environment. And we have an exciting lineup of speakers to talk to us about how the chemical components found in our food and the environment could be stimulating different biological responses in humans, particularly the biological pathways associated with food allergies, and that may contribute to the development of allergic diseases. So join us monthly for these seminars, and we'll spotlight the critical science each of these researchers and experts is doing. And today we are delighted to be joined by Noah Whiteman, professor of genetics, genomics, evolution and development, and the director of the Essig Museum of Entomology at UC Berkeley. So Dr. Whiteman will discuss his book, The Most Delicious Poison, Story of Nature's Toxins from Spices to Vices. And then at the end, we'll talk about how that research really links up with FOSSE's work to identify these adjuvants and understand how they impact human biology. Unlike typical seminars, instead of doing a full-on conversation back and forth the whole time, I'm going to give Noah the mic to talk to us about this great research he's put into the new book. So without further ado, thank you for joining us, Noah. And
my pleasure.
And there'll be questions. Put your questions in the Q&A box, and we'll get to those at the end.
Great. Can you hear me okay, Megan?
Yeah, you sound great.
Thanks for that introduction, and thanks to your institute, the FOSSE Institute, and the listeners, people watching for joining. It's 9 o'clock in Oakland, California. It's noon there, so some of you are probably eating lunch. And this is the perfect time to take a look at a big picture view of evolution and our interaction as animals with the chemicals that other organisms make that have become so important in our diet and in our health. And I use the word evolution because I'm an evolutionary biologist, and that's the perspective that I will share with you today. And taking a very big picture perspective, this is a view of underneath the rings of Saturn, the gas giant planet. And this was taken by the Cassini spacecraft to recreate this famous pale blue dot photo that Voyager 1 took that Carl Sagan had set up. So that little dot there is Earth, that's us. And there's that famous quote from Carl Sagan. See here if I can get this to work. There we go. Look at that. Look again at that dot. That's here. That's home. That's us. So as I walk you through this talk, I want you to think about yourself. If you were a visitor to our solar system looking for life, you're going past these big planets. Yeah, maybe Europa has some microbes under its frozen ocean. Who knows? But the planet where the real action is the Earth. And life as we know it is quite remarkable in how bellicose the interactions are between species. But before we get there, as you were approaching Earth, you would start seeing, this is that famous Earthrise, taken in the right context here, as Earth was coming out from the side of the moon by Bill Anders. And you can see the beginnings of the South American continent here, and it starts to look green. So even though our world is a water world, which is why it's so blue in part, there's a lot of land. And on that land, most of it has been colonized by land plants, which are, of course, derived from algae that lived in water about 500 million years ago. And once plants colonized land, they kind of took over. And so this is a picture of the Amazon basin and the Andean mountains of South America. And so as you were coming in, say your alien spacecraft sent a probe in to check things out, this is how it would begin to look. It would get greener and greener, but you would start seeing river valleys, things like that. This is the Amazon basin. And your spectrophotometry, your photographs would start appearing bright green. And as you got even closer, you'd start making out the crowns of individual trees, and you'd see a lot of diversity along the equator, where the vast majority of life occurs. And the vast majority of species of life on Earth occur on land, not in the water. And they occur in the region between the Tropic of Cancer and the Tropic of Capricorn, the vast majority of species. And what you would see if you zoomed in even further would be insects, some other non-insect arthropods like mites, and you would see insects that are mainly eating living plant tissue and fungi as well, but it would mostly be living plant tissue. And these insects are the species that I study, their interactions between them and plants. Here's a picture of a monarch butterfly caterpillar from my backyard that's feeding on a milkweed. So this wouldn't be from the Amazon rainforest, but these other species are just a mix of the kinds of species that you would see. And so the most species of life on Earth with names are herbivorous insects and the plants that they eat. So those two groups of life account for more species with names than any other. Even though there may well be more microbial species, they're harder to describe and harder to find. And so the macroscopic life on Earth is most of it is either a plant or an insect that eats the plant. And we think that interactions between these two lineages, between insects and plants, are what drove the diversification of both sides of the interaction through a series of chemical arms races that plants evolved the ability to protect themselves, not only with things like thorns and hairs that have venom, like stinging nettle, but also with a myriad of specialized metabolites that they have evolved over the last 500 million years to defend themselves against enemies like these insects. But nature finds a way and the insects evolve a way around that eventually. The trade-off is that they often become specialized on a particular set of plants that make a particular set of specialized metabolites. Like the monarch butterfly can only feed on plants that make cardiac glycosides in the milkweed family, which are normally well protected from other herbivores. So this balance, this trade-off, this dynamic, this coevolution between plants and animals is what I study. Often the animals themselves, the insects, will take the chemicals made by the plants that are toxic and they evolve resistance to them, and then they use them for their own devices. So the monarch butterfly is brightly colored here, the caterpillar and the adult, because they're sequestering the heart poisons from the milkweed to protect themselves against birds. So the bright colors were selected for by the bird brain to warn the birds not to eat them. The butterfly wins and the bird wins. And if indeed a bird did eat, say, 3 or 4 monarch butterflies, it would probably die from cardiac glycoside poisoning. So what I want you to think about is whether and to what extent we're really that different from these animals that are interacting with plants on a daily basis. So plants, as I said, they're not sitting ducks. This is a sped up video of a special kind of microscopy of a plant called Arabidopsis thaliana, which happens to be the genetic model plant that we use to study plant biology. And there's a caterpillar you can see very quickly in the bottom part of that screen that's eating the leaf. And you can see the plant is lighting up. And this plant is a genetically modified plant that has been made so that we can see a message that is sent between cells via calcium. It's actually glutamate-mediated calcium release. And what's happening is when the plant senses that it's being damaged by the mandibles of that caterpillar, the plant sends this message quickly across its phloem tissues, the sap tissues, so that leaves that are distant from the wound are actually have a signal that they receive that allows them to turn on their anti-caterpillar defenses, even in leaves that are not being attacked, so that they can anticipate attack by that same caterpillar. So the message here, this is my favorite New York Times article. Sorry, vegans, Brussels sprouts like to live too. And so this is just to remind us that plants, are not here for us. They evolved because they have their own interests in reproducing and surviving, like anything that is bound by evolution by natural selection. And they've evolved myriad ways to defend themselves. And that has been both to human benefit and human harm, as you'll see. And like all of these things that didn't evolve for us, we have to take each one as they come and remember that there is no design. These things were not made for us. Yes, we have intervened and we have tapped into these resources, these metabolites for better and for worse. And in the case of food crops, we've domesticated plants. But in many cases, we have left the specialized metabolites there, presumably because they protect our crops from attack by natural enemies. These are just some of the chemicals that specialized metabolites that plants make that they don't need in order to grow and reproduce in the absence of enemies. So when scientists first found these, they're like, what are these? Are these storage molecules for nitrogen? No, is the answer. And you'll see that what they evolved to do for the plant is usually to protect them or somehow manipulate the animal mind. The one on the left is strychnine, cocaine, DMT, THC, cinnamaldehyde, which gives cinnamon its spice, huperzine A, which is actually from a club moss and is a sort of emerging drug. Taxol, which may have saved some of your lives, the anti-cancer drug, which is from yew trees. Alpha-pinene, which I'll talk about in a minute. Caffeine, which I'll talk about after that. Myristicin from nutmeg, which I'll talk about almost at the end. And at the very end, I'll talk about morphine. These four, just to give you a vignette of walking kind of through the threads of the book. And so I have a chapter on the most diverse class of specialized metabolites in the natural world, which are terpenoids. And these have an isoprene backbone. That's what defines them. They're very diverse and abundant. And this quote by de Candolle kind of sums it up. He was a famous biogeographer of plants. And he said all the plants of a given country, all those of a given place, are at war with one another. And what he meant by that is plants are competing for resources. They're competing for sunlight, soil, water, and any advantage that gives them a leg up over the competitors, will evolve and spread in populations. And those include defenses against natural enemies that are taking away their capacity to grow and compete. And in this chapter, I talk about ending up in a coast redwood forest above my house in the Oakland Hills. And here's a picture of redwoods and a person standing under them, and taking a deep breath to calm myself in a particularly stressful period of my life. And as we all do in the forest, you have this sense of, oh, this is a really complex bouquet of stuff I'm inhaling. And usually it's a good set of smells. You have a positive feeling about that. And one of the terpenoids that you would inhale in most forests is something called alpha-pinene, which you might also find in a diffuser or a scented candle. And what's amazing is that the target of that alpha-pinene in the animal body is something called the GABA A receptor. And this is the same receptor that Ambien binds to. And so plants have evolved this alpha-pinene for myriad reasons. One of them is to protect themselves against natural enemies like insects. And if an insect feeds on alpha-pinene, my lab and others have studied and showed that alpha-pinene is an insecticide because it binds to the insect's GABA A receptor in the brain and slows them down. And that's good from the plant's point of view because they're eating less. And so plants are making these terpenoids in part to protect themselves from enemies. It's binding to the same ancient receptor in neurons that when they fire, they put your brain to sleep. And so the GABA A drug Ambien acts very much like alpha-pinene, even if it binds to a slightly different part of the GABA A receptor. Here's a fossil, just to show you how ancient these interactions are. This is the oldest known fossil of plants in the same genus as redwood, sequoia. This one's from Inner Mongolia, and it's very old. It's about 170 million years old. But if you look at the needles, if you've ever seen a coast redwood, it looks just like a coast redwood now. So there's been a remarkable degree of morphological conservation in the evolution of these plants. And what I love about this fossil bed is that there are insects that are preserved right alongside these fossils. And that's what this thing is over here. It's a plant-feeding insect in the order Hemiptera that itself is 170 million years old. And the more remarkable thing about all this, circling back to our original slide, is that the rings of Saturn are actually younger than this fossil. So the rings of Saturn are thought to be about 100, 120 million years old. We know that from Cassini's work, actually. And so the sequoia lineage and the insects that have been attacking it are older than those rings. So these dynamics are ancient. They've been going on for a long time. And we have to remember that when we tap into this, this sort of arsenal of chemical defenses that we use and abuse. Perhaps the most important set of chemicals from a human kind of drug side are the alkaloids. And these are chemicals that often have a nitrogen in their structure. And one of the most favorite ones is caffeine, especially of mine. And caffeine, of course, is the reason that most of us drink coffee or tea. And caffeine itself has evolved in plants at least 6 times independently. We know the genetic basis of that. It's kind of remarkable. It turns out that caffeine is a natural insecticide, and this was work done in the 1980s by James Nathanson. And so coffee and tea plants are what he studied. And in a series of experiments, here he sprayed tomato leaves with caffeine and placed a caterpillar called the tomato hornworm on them. And this was just water. And you can see that the caterpillar liked the leaf so much. You just see the mid vein of the leaf, the midrib, and then you see a chrysalis here of the caterpillar. So it liked it a lot. When you spray 0.1% of caffeine, you can start seeing the caterpillar didn't complete development, but it ate most of the leaf still. Here's 0.3%. It eats less of the leaf, and there's a smaller caterpillar up here. And then if it's 1% caffeine, which is the beginning of what you would see in tea leaves and caffeine coffee beans, you can see the leaf is mostly intact with some nibbling, 3%, a little less nibbling even. And in both cases, all of these caterpillars died. And so the caffeine is binding to the same receptors in the caterpillar as it is in our brain, but they're little, and the dose makes the poison in this case, and they are eating a lot of it when they're taking a nibble of that coffee bean or tea leaf. But it's not only as sort of a defense that plants use caffeine. Citrus plants and coffee plants put caffeine in their nectar, and they've evolved to do this, we think, to manipulate the minds of pollinating insects like bees. Because when they do that, they increase the ability of the bee to remember that plant through an odor-based memory and return to it, to the detriment of the bee, because at some point they're losing out. They're not getting the pollen that they want. They're not getting as much nectar as they want. But the plant is getting pollinated for sure by doing this. And so this was a paper published in the journal Science that was showing that plants put caffeine at levels in the nectar that's just below the bitter rejection threshold that bees have when they're tasting a substance. And it's in some cases about the level that you'd find in instant coffee. Usually it's a lot lower than that, but it's still enough to cause an enhanced memory reward. So it's not just a defense that plants use these chemicals, they're also using them to sort of for their own way to manipulate insect minds to increase their fitness, not just avoid attack. But it all comes down to us. And this is a very interesting long-term study that I should mention was not an experiment. So that means we can't separate cause and effect here, and we cannot eliminate the possibility that people who drink a lot of caffeine or coffee or tea, that there's something else about them that causes them to have reduced depression risk. At the same time, this is what the data show. So this was a very large study of about 51,000 women nurses. This was done over a 10-year period, tracking them and measuring depression symptoms. None of the women had depression at the beginning of the study. And what they found is when they tracked the amount of caffeine consumption, that there was a 20% on average reduction risk in women who drank roughly 3 to 4 cups of coffee a day compared to control. And they did not find this pattern in women who drank decaffeinated coffee. So again, it could be something about people who drink decaffeinated coffee that increases their depression risk, irrespective of the caffeine, or it could be the caffeine that is driving this. And so a similar study that was about double the sample size that involved men, and it was a physicians and nurses study, found that there was a 50% reduction risk in suicide over a 10-year period. And of course, there were far fewer suicides than depression risk, thankfully, or depression episodes. So the power is a little lower in some statistical sense, but still, this also suggested that there's at least a correlation between the amount of caffeine consumption up to a point and suicide and depression risk. If you go over 4 cups a day, the depression risk starts going up and other things start happening. So this is another example of it's the dose that matters, of course, for all of these pharmacological effects. And sometimes it's too much. Like this very sad situation with the 17-year-old from Australia who died of a caffeine overdose when his friend gave him a packet of powdered caffeine. He put it in his protein shake before he went to bed and drank it and didn't wake up. And so, the amount of caffeine that can kill you is surprisingly low. And so this is something to keep in mind, that it's very dangerous to use powdered caffeine. Even caffeine tablets that have gelatin, that are gelatin coated, can be very dangerous if used in abundance. So this is something that, like all things, this two-sided nature, we have to keep in mind. So the other pseudoalkaloid I wanted to tell you about today, the second to the last one, is myristicin, which gives nutmeg its spicy, wonderful flavor. And it turns out that myristicin, which is of course in the seed of the nutmeg, you can see in this nutmeg that this bright orange net-like thing is actually called an aril, and that is where the spice mace comes from. So nutmeg and mace come from the same plant, from the same fruit. It's just that nutmeg is a seed. And that myristicin, it turns out, is a potent insecticide. And this is just showing that fruit flies die in a very dose response positive way when they're exposed to myristicin in the diet, which may help us understand why the nutmegs make it in the first place. Probably protects the seeds from things like weevils. Nutmeg can also kill people. People misuse nutmeg as a hallucinogenic. Don't do this. In this case, there was a woman who died of a fatal overdose, and the reason that the physicians who were doing the autopsy discovered this is they removed her stomach and they could smell the nutmeg. There was so much in her body. And so this is also showing that you may have heard nutmeg, can get toxic at doses again that are not as high as you would think. So this is just yet another example of these things didn't evolve for us. They're not made for us. We have to remember that whenever we're using these chemicals. And then to tie this into the kind of grand arc of the story of nature's toxins, the story of nutmeg and myristicin, this one alkaloid or pseudoalkaloid, plays a particularly important role in the last 500 years of human history. So nutmeg originally was native and endemic to the Banda Islands, which were known as the Moluccas in what is now Indonesia. It's the only place in the world they grew. Now they grow in many places where the trees have been moved. And the Spice Islands, as they were called, and the Banda Islands in particular, were the subject of repeated colonizations by Western Europeans that started with the Portuguese and ended with Dutch rule, but in between, the British had control of them. And there was a treaty signed in 1667 where the last island controlled by Britain was the island of Roon here in the west. The rest were controlled by the Dutch. And to end this war and other wars that these countries were engaged in, they signed this treaty in 1667, which gave the island of Roon back, or to the Dutch, I should say, in exchange for what is now, part of New England, which includes New York. So you can think of this pursuit of this alkaloid, which was so expensive and coveted and more expensive than gold per weight in Europe, that drove this major geopolitical transfer of power and land. The problem was Britain was kicked out of the spice trade as a result of this. But the other issue that they had was Britain was now addicted to caffeine and tea that they were importing from China and paying for in sterling. So they had this huge trade deficit that they could no longer offset with their spice trade. But what they did have was control of the poppy fields in India that they were in control of, and they began to import opium illegally. Oh, and by the way, this is just showing that flies hate morphine. This is another alkaloid that is from the poppy plant, from opium. And probably this evolved as an anti-herbivore chemical as well. And over time, from 1650 to 1880, there was an exponential increase in the amount of illegal opium smuggled by the British into China. And it resulted in about a third of the adult male population of China probably having something called opium use disorder. And this is showing just a preparation. All of these pots have opium in them in India, and they're going to be brought to factories, ports in the Pearl River, and smuggled into the interior of China. And this allowed Britain to rebalance its trade deficit to the great detriment of China, which was already undergoing a lot of internal strife. But this didn't help the fact that now all of a sudden about a third of the population of men are addicted to opium. And so eventually the emperor decides, really, this is enough. And they really ban it, and they start dumping out the opium into the river. And the British didn't like this. They sent a fleet of ships into the mouth of the Pearl River and began the Opium War, which eventually resulted in a very lopsided treaty that gave Britain Hong Kong permanently, at least until 2 decades ago. And it really reset our whole geopolitical order. and eventually was one factor in leading to the rise of modern China. So we have the war of nature, the war within, and the war of men. And that's what my book is about. That's what I'm fascinated by. And I know this was a little bit of off topic for you, but I hope it helped you understand a little bit more about what's really going on that pale blue dot, which is, you know, a very bellicose set of interactions between species that looks placid from the outside, but it is not, is anything but. So thank you very much.
This is wonderful. No, and I disagree that it's off topic because these secondary metabolites that you study and that you're researching are so impactful on our health, and we're just beginning to understand what's going on there. That's what our seminar series is all about this year. So there's the book if anyone wants to pick it up, and you can wrap up your slide sharing, and Julie will put up. So folks, we'll get to Q&A in a moment, but real quick, I want to show people often ask how they can get more involved with FOSSI. Scan and get in touch with us. People donate, they host events, they follow us on social media, and keep in touch with us via the seminar and email. So next month, November 12th at noon, we're going to have Thomas Hartung, a doctor, MD, PhD, who's going to talk to us even more about food chemicals from the toxicity side specifically. So let's talk Q&A. So to get us started, I got to pick some questions to start with. So thinking about FOSSI's SAFE program, these secondary metabolites, natural or synthetic chemicals even that have an impact on food, from our food and environment that have an impact on atopic disease and food allergy. In your research, do you also see these secondary metabolites having an impact on the onset of chronic disease? Can you tell us about that?
Yeah, so my lab does very basic research, and so we don't really study human disease. But one of the model systems that we use is a naturally co-evolved system that involves plants in the cabbage family. So things like broccoli, cabbage, kohlrabi, radish, watercress, Brussels sprouts, kale. You get the idea. The things that have a peppery or sulfur kind of smell to them. So those chemicals that are producing that smell or the peppery taste start with a specialized metabolite called the glucosinolate, which is made by these plants and all their tissues. And then when an animal chews on the leaf, there's an enzyme that's in a different part of that plant cell that combines with the glucosinolate that the plant makes and creates this mustard oil bomb that in your mouth, you're making that by chewing the leaf up. And that's doing just as the plant wants when all is lost. And it's basically trying to create this aversive reaction that prevents you from eating more. So your wasabi receptor in your mouth that detects this spice is triggered. And so, but the funny thing about us is that many of us like that. So at some point we start eating more of it, right? But children don't like it. That tells you something about probably how that evolved, right? It evolved to prevent things from eating it. But the chemicals that are the downstream products of those that cause the mustard oil bomb to go off are called isothiocyanates or mustard oils. One of them is called sulforaphane. That is one that is most abundant in broccoli, for example. So if you eat a raw broccoli floret, you're getting a lot of sulforaphane. And that's a toxin. So in cells, it would damage cells, but a little bit, the amount that you would get when you eat, say, a serving of broccoli, your body's able to handle that so that it's not really very toxic. And instead, what happens is it turns on a normal detoxification response that's called the mercapturic acid pathway. The chemical glutathione, which your cells make to sort of bind to toxins, and then there's an enzyme that catalyzes that called the glutathione S transferase. Those get turned on, those pathways and those enzymes, the glutathione S transferase enzymes, and that probably allows your body to detoxify other things that are around because they're very promiscuous enzymes for the most part. And so there may be this spin-off positive effect of turning on this mercapturic acid pathway. So I became most intrigued by this by research that other labs have done that started with fruit flies, Drosophila, the genetic model animal. And what they did is there's a Parkinson's model of fruit flies that actually has the human Parkin gene inserted into its genome. And that Parkin gene causes one of the causes of familial Parkinson's, which is a rare form of Parkinson's, but is easier to study because there's a simple genetic basis to it. So these fruit flies are expressing the human Parkin gene, and they have tremors. They have symptoms of a Parkinson's human patient. And what scientists found was when they fed the flies sulforaphane that had the Parkin gene, or a control diet that didn't have sulforaphane, those flies were really prevented from developing symptoms of Parkinson's disease. And the scientists proved that it was this mercapturic acid pathway that was the reason why. And that's because there are mutant fruit flies that don't make glutathione, or they don't make glutathionase transferases. And so this was amazing. And it suggested that, maybe this diet in fruit flies could prevent the progression of Parkinson's. So then that study was repeated in rodents, and there were no genetic model Parkinson's available. So what they did is they did a chemical sort of mimic of Parkinson's by injecting a toxic form of dopamine into the neurons in the brain that start dying in Parkinson's patients to mimic the progression of Parkinson's. But they first gave the rodents sulforaphane in their diet, in their food pellets, or not. And they found the same thing that they found in the fruit flies, that the progression of Parkinson's was really slowed in the rodents that had been fed this sulforaphane. Now, people have tried doing similar studies in humans, but the problem is they were done in patients already having Parkinson's, and it didn't seem to help them. But the question about prevention is the critical one here, I think. So that's maybe the most interesting one, where you get this thing it's a toxin, isothiocyanate. Most of us wouldn't think of wasabi as that, but your body is telling you this. And you're crying after you eat wasabi, right? So, but at the same time, we're very large animals, but in the grand scheme of things, we're able to handle a lot of these specialized metabolites. So, and then the slight cranking on, which is called hormesis, is a general phenomenon, may in fact have these spin-off knockoff effects.
Definitely. Well, that is wonderful. And I could talk about this all day with you, but I want to let people go. But I hope you have a great book tour. Thank you so much for sharing this great science with us. And thank you to everybody watching at home. We look forward to joining us again on November 12th. Thanks, everybody.
Thank you so much for having me.